Hi, Assalamualaikum. We are from Group Gryffindor from AS1203B with our case study number 2, which is about artificial blood. And this is our group members, Arwah binti Muhammad Yusof, Nur Husni binti Bahari, Fairuz Niza Nur Ain binti Jamal, and me, Nuri Madiana Humaira binti Ruslan. Our case study questions are: how do scientists make artificial blood and the effectiveness of the artificial blood compared to the real blood? And our objectives for this case study are to determine the definition of blood and artificial blood. Second, to study the process of making artificial blood. And lastly, to differentiate between real blood and artificial blood.
Assalamualaikum warahmatullahi wabarakatuh. My name is Arwah binti Muhammad Yusof. I'm from class AS1203B. My student number is 2020840396. Okay, now let's move to the introduction of this case study. First, what is blood? Blood is something that are essential for all living things. It made up of liquid and solids. For liquid part, it's called as plasma. Plasma is made of water, salt, and proteins. Over half of blood is plasma. For the solid part, there are three components, which are red blood cell, white blood cells, and platelets. The function for red blood cells is to deliver oxygen from lungs to organs. For the white blood cells, the function is to fight infection and as a part of immune system. Lastly, for the platelets, the function is to help blood work around a cut or wound. Okay, the plasma, red blood cells, white blood cells, and platelets are also the four components that comprise human blood. Blood also transport essential substances such as oxygen and nutrients to the body cell. At the same time, it also transports metabolic waste product away from the same cells. Blood is pumped by heart to all parts of the body. Okay, next, what is artificial blood? Artificial blood is a theoretical substitute blood product that can carry out the most vital function of blood, which is the transport of oxygen and carbon dioxide. It also could be a life-sustaining measure, especially in times of serious blood loss during large disaster. However, the product in development right now cannot carry out the secondary function of blood, like fighting infections.
Next, the history of artificial blood. Actually, medical practitioners has tried numerous substances like saline, milk, plant resins, urine, and also blood sheep as a substitute for blood. In 1854, milk was injected in patients to treat Asiatic cholera, which physicians believed that it helps to regenerate the white blood cells. Next, the possible substitute in the history is saline solution, which the experiment was done on frogs. The scientists believed that they could keep the frogs alive if they remove all of the blood from the frogs and replace it with saline solution. However, the results were a little misleading because it later determines that the frogs can survive for a short period of time without any blood circulation at all. During the 18th, other materials like hemoglobin and animal plasma was discovered to be utilized as blood substitute. However, it was hindered by significant technological problems. Next, how do scientists make artificial blood? The materials that being used in artificial blood are perfluorochemicals, PFC, hemoglobin-based oxygen carriers, HBOCs, raw materials, and also hemoglobin substitute. Next, PFC. PFC are biologically inert materials that can dissolve 50 times more oxygen than blood plasma, and they are also inexpensive to produce and free of biochemical materials. So, this eliminates the possibility of spreading infectious disease via blood transfusion. The problems with PFC are it is not soluble in water and it have ability to carry less oxygen than hemoglobin-based product. So this means that more PFC must be used. Improved PFC emulsions are being developed, but it have yet to reach the market. Next, hemoglobin-based oxygen carriers, or can be called as HBOCs. HBOC utilize the same oxygen carrying protein molecule found in blood, which oxygen bonds covalently to the hemoglobin. This hemoglobin product does not have membrane, so the problem of blood typing is eliminated. And also it can be stored in a long period of time. And down here is the simplest process of making artificial blood. Synthetic hemoglobin products are produced from hemoglobin that harvested from E. coli bacteria strain. And then after 3 days, the hemoglobin grown in the seed tank and then it was fermented. Next is the hemoglobin substitute, which means that it needs to modify the hemoglobin molecule. This process involves chemically cross-linking molecules or using recombinant DNA technology to produce modified proteins. Intramolecular cross-linked hemoglobin are not significantly increased in molecular weight, but have specific chemical cross-links between polypeptide chains to prevent dissociations to dimers or monomers. These modified hemoglobins are stable and soluble in solutions. Theoretically, these modifications should result the products to have greater ability to carry oxygen. Next is raw materials. To produce hemoglobin synthetically, manufacturers use compounds known as amino acids. There are 20 naturally occurring amino acids that may be used to produce hemoglobin. All of the amino acid group share certain chemical characteristics. There are carboxyl group, a amino group, and also a side chain which it differentiates the various of amino acids. Hemoglobin synthesis also require a specific type of bacteria and all of the materials needed to incubate it. And the materials needed are warm water, molasses, glucose, acetic acid, alcohol, urea, and liquid ammonia. Next, the process of making hemoglobin synthesis to produce artificial blood. A strain of E. coli bacteria is used to make hemoglobin. About 3 days, the protein is harvested and the bacteria are destroyed. To start the fermentation process, a sample of the pure bacterial culture is transferred to a test tube that contains all the nutrients necessary for growth. When the population is great enough, they are transferred to a seed tank. A seed tank is a large stainless steel kettle that provides an ideal environment for growing bacteria. It is filled with warm water, food, and an ammonia source, and also vitamins, amino acids, and minor nutrients are also added in the tank. When enough time has passed, the contents of the seed tank is pumped to the fermentation tank. The fermentation tank is a larger version of the seed tank. It is also filled with a growth media needed for the bacteria to grow and produce hemoglobin. Ammonia water is added as pH control is vital for optimal growth. And when enough hemoglobin has been produced, the tank is emptied so isolation can begin. And then, the hemoglobin is transferred to a final processing tank. In the final processing, the hemoglobin is mixed with water and other electrolytes to produce the artificial blood, like in the figure at the right. The artificial blood can then be pasteurized and put into an appropriate packaging. The quality of compounds is checked regularly during the entire process. Frequent checks made on the bacterial culture are also important. Besides, various physical and chemical properties of the finished product are checked, such as pH, melting point, moisture content, and etc. This method of production has been shown to be able to produce batches as large as 2,640 gallon or 10,000 liter.
Okay, we will see about the difference between real blood and artificial blood. For real blood, only have life force of 42 days with refrigeration, while artificial blood have some life force of 1 to 3 years without refrigeration. For real blood, they have a group A, B, AB, and O, while artificial blood only have universal group O negative. For real blood, patients would receive serious health problems if they receive incompatible donated blood, but for patients who receive artificial blood will not experience immunologic reaction. This is because artificial blood is compatible with all types of blood because artificial blood only have universal group O negative. Okay, for real blood, it have the possibility of contaminated with HIV and hepatitis virus, while manufactured artificial blood can be sterilized to destroy bacteria and virus, so they are immune. And real blood may not be readily available when needed in emergency situation, while artificial blood can safely shelf outside hospital and then rapidly administer to patient in emergency situation. All in all, this can show artificial blood is much more effective and advantageous than the real blood.
The conclusion of the artificial blood is the real blood consists of cell and platelet that suspended in the plasma. Plasma protein is influence the blood pH, osmotic pressure, and viscosity. It also functions in lipid transport, antibodies, and fibrinogen. A blood artificial is important to replace as a red blood cell when human lost a lot of blood. Blood artificial have the same function as red blood cell to transport oxygen and carbon dioxide throughout the body. The benefit of blood artificial, it can be safely shipped out hospital and rapidly give to patient in emergency situation. The disadvantage of blood artificial is it can only carry the oxygen and carbon dioxide and cannot carry other nutrients through the body. It also can cause the hemosiderosis and chronic overload. Research on artificial blood has not led to an approved medicine in the market today, but the progress has been made since 1978. For the first time, clinical trial of HBOC was carried out. The new technology has been released to create the fake blood and it gives same function in human body, but it also cannot be the real blood as well. So, we need to practice a healthy lifestyle and eat foods that replenish red blood cells such as red meat, organ meat like liver, beans, legumes, egg yolk, and dry fruit such as prawns and raisins. That's all from me.